Hi everyone, this is Adriana from DocBL, and I'm here to talk to you about cancer genetics. So what is cancer? Cancer is the uncontrolled cellular growth. But how do you get uncontrolled cellular growth? You have to have a mutation in a gene affecting the growth of a cell. So first, let's talk about how we get mutations. DNA polymerase is responsible for DNA replication, and it does a really good job, but sometimes it makes mistakes. These mistakes are fixed by DNA polymerase's proofreading abilities. After replication, the cell has its own way of checking for errors, and that's through mismatched repair mechanisms. But of course, replication errors aren't the only way you can get a mutation. They can also be spontaneous, so that means that they just happen randomly. And a good example of that is bcr-abl, but we'll talk more about that later. Chemicals such as cigarette smoking, radiation like X-rays, infection like HPV, human papillomavirus, inflammation, and inherited mutations like breast cancer. So, we remember that we said that cancer was caused by having a mutation in not any gene, but a gene that affects the growth of a cell. Broadly, these genes are separated into oncogenes and tumor suppressor genes. A tumor suppressor gene is a gene that indirectly affects cellular growth, and so when it's mutated, it's called a tumor suppressor gene. And this mutation leads to a loss of function. It's inherited recessively. This can be explained by the two-hit model. So we have two copies of every gene. So if you have two good copies, then you're okay. If you have one bad copy and one good copy, you're still okay because your good copy takes over. But if you have two bad copies, then you're in trouble. So let's take the example of retinoblastoma. This is a tumor of the retina. Retinoblastoma is also the name of a protein that monitors the cell cycle at G1 phase. So if a cell has damaged DNA, it can't get past this to proliferate. Some people inherit one bad copy of this RB gene, and that means that they are 6 times more likely to have cancer because they just need one more mutation for cancer to be caused. A very common tumor suppressor gene is p53. It is also known as the guardian of the genome. p53 is a transcription factor, and when a cell is acting abnormally, it stops it. So if a cell has damaged DNA, p53 stops it and causes it to apoptose, which is cellular suicide. However, if p53 isn't working properly, that can lead to abnormal cell growth. Now moving on to oncogenes. Oncogenes are a little bit different than tumor suppressor genes. They're more like having the accelerator pedal stuck down. So they're a gain of function mutation. An example is RAS. RAS is switched on by cellular growth signals, and it stimulates cellular growth. There are different mutations that can cause RAS to be overstimulated. Another oncogene is the bcr-abl. So this is less of a mutation and more of a chromosomal abnormality. This is when part of chromosome 9 and part of chromosome 22 fuse together to form the Philadelphia chromosome. This new protein that's formed is a tyrosine kinase that leads to uncontrolled cellular proliferation. Another oncogene is MYC. This one operates a little differently. A transcription factor causes it to be transcribed, and then the MYC protein leads to cellular growth. A mutation in the transcription factor means that there's lots of transcription factor that stimulates lots of MYC growth, and that leads to abnormal cellular growth. Okay, so in review, cancer is uncontrollable cell growth. Tumor suppressor genes are like having bad brakes, and their inheritance can be described by the two-hit model, and examples are retinoblastoma and p53. Oncogenes can be described as having an accelerator pedal stuck to the floor, and they are gain-of-function mutations, and examples of these are RAS, bcr-abl, and MYC. So that finishes our cancer genetics lecture. Thank you very much.